Hello, everybody. My name is Stephan. I'm educator at the Serengeti-Park and chair of the second session. Our next three talks will focus on comparative studies in primates. And we directly start with our first speaker. It's Dr. Katrin Kopp from the Max Planck Institute of Evolutionary Anthropology. And she will talk about behavioral and cognitive studies on zoo-housed great apes. And she pre-recorded her talk for us. So Friederike, please, can you start the video?
Thank you very much for your kind introduction and for inviting me to this conference. What I like to do today is to illustrate how zoo studies can contribute to our knowledge about great apes in a very particular way, and how zoo studies can support efforts to increase the welfare of apes in human care, even when they are not explicitly welfare studies. And I'd like to take the opportunity to thank all the colleagues at the zoos and sanctuaries I have the pleasure to work with. The title of this talk, Great Apes Are Not All Alike, has more than one meaning. The most obvious one, apes belonging to different species are not alike, but also apes within a species are not all the same. And considering both levels is relevant not only for science, but also for husbandry and zoo management decisions. And today I want to address both perspectives. The first part will focus on food sharing in zoo-housed orangutans compared to chimpanzees and bonobos, while the second part will focus on inter-individual variation in zoo-housed chimpanzees. And let's start with the orangutans. My research on zoo-housed orangutans started 11 years ago in the ape house at Berlin Zoo, when I observed an adult orangutan male passing over food to his immature daughter. And the question popped up, why did he do that? In the wild, he wouldn't have the opportunity to share with his offspring. By that time, food sharing in chimpanzees and bonobos, the closest living relatives to humans, was already well studied in both wild and zoo populations. In both pan species, the apes share with offspring, like in many other primate species, but they also, unlike most other primate species, shared among adults. There is regular sharing of meat and large fruit, and selective sharing with valuable partners. Moreover, bonobos even share food between groups and with unfamiliar conspecifics. Food sharing can serve several functions, from the nutritional one to harassment avoidance to reciprocating favors, and related to this, to establishing and strengthening social bonds, which is a pillar of cooperation and therefore important for cooperative species. On food sharing in orangutans, on the other hand, was much less known then. Field studies reported that mothers frequently share food with their dependent offspring, which most probably facilitates social learning about food and how to process it. Among adults, however, food sharing occurs only occasionally, mostly between potential mating partners. Until then, no systematic study on food sharing had been published, so the question whether food sharing in orangutans was similar or different from chimpanzees and bonobos was still open. Orangutans are special in many ways, but particularly in their social organization. In contrast to bonobos and chimpanzees, they do not live in big groups, but have a semi-solitary lifestyle, with mothers and their dependent offspring as strongly bonded, long-lasting social units. In contrast to pan, orangutans don't hunt, nor are they regularly engaged in coalitions or border patrols like in chimpanzees. So, apart from the mother-offspring context, there is barely the necessity or even the opportunity to share food in orangutans. In the wild. In zoos, the picture is a completely different one. Here, the orangutans live in permanent groups where serious conflicts are rare, and when they occur, they are able to manage conflicts, for instance by a third-party intervention or reconciliation. And even adults play with each other. So they are not per se our unsocial cousins. But how might these two pictures, how might this flexibility in social behavior be explained? I'd suggest that orangutans have a social behavioral potential that is similar to that of other great apes. And despite fewer occasions in the wild, it is utilized when living in permanent groups. If that is true, the food sharing patterns in orangutans might also be similar and serve similar functions as in chimpanzees and bonobos. And to investigate this, Katja Liebal, who was my PhD supervisor then, and I conducted a food sharing study with Sumatran orangutans at 2 German zoos. One group of 4 individuals in Berlin and 2 groups of 3 individuals, each in Dortmund. The latter were housed in 2 adjacent indoor enclosures during the winter, but joined each other in the summer in the outdoor enclosure. But also indoors, they could interact with each other through the separating mesh, which they actually did quite frequently. We tested each group on 11 days, and the test setup was very simple and comparable to chimpanzee studies using monopolizable food. For each session and group, we put a large and compact fruit, a pumpkin, a melon, or a pineapple into the enclosure. Then the orangutans got access, and we started to video record all the interactions over food until food was up or ignored. Subsequently, we analyzed all food interactions between a possessor and solicitor. To test whether the relationship quality affects the probability of sharing, we calculated a proximity-based sociality index as a proxy of relationship quality for each direct diet. This so-called possessor-centered sociality index, PSI, is ego-centered and accounts both for the possessor's perspective and for the fact that individuals considerably vary in their general gregariousness. Then we coded the food interactions, which are interactions over defensible food between a possessor and a non-possessor. A food interaction has 3 possible outcomes. It may result either in no food transfer at all, or in a non-tolerated transfer when food was taken by force, or in a tolerated transfer. And only tolerated transfers are called food sharing. Food sharing can be either passive or active. In passive transfers, the possessor only tolerates the taking of food. And in active transfers, on the other hand, the possessor is actively involved in the process. Most food sharing Now I'd like to show you how it looks like when orangutans share actively. And first, you can see a transfer between 2 unrelated females. The one on the top is the possessor. Now you can see an interaction between the male and the female. The female first takes a bit herself. It's passive sharing, but she's not that successful. And now the male bites off a piece and give it to her. And again, another male, another female sharing actively food. And here is a very special situation. In one zoo, the 2 groups were separated but housed next to each other in the winter, and there were several interactions and food sharing through the mesh, and here's one of them. You can see a nice begging gesture, which is successful. Coming to the results. In this figure, the y-axis indicates the number of food interactions. The right column shows those that resulted in food sharing, and the left column shows those that did not. In total, we saw more than 700 food interactions, of which a remarkable proportion of 58% resulted in food sharing. We observed all types of tolerated transfers, passive and active, passive shown in green, active in blue. And compared to reported proportions of active transfers in chimpanzees and bonobos, and also in wild orangutans, 29% is a lot. Furthermore, forced transfers or conflicts about food were really rare. The far majority of food interactions, whether they resulted in sharing or not, was peaceful and it included many communicative signals like begging gestures. As pooled data say nothing about individual interactions, here's a network graph for all three groups. Circles indicate individuals, arrows between them show food sharing, directed, of course, and the size of the circles indicate the time this individual had been in possession of the food source. The bigger the circle, the longer this individual was the food possessor. And the width of the edges indicate the number of tolerated transfers from one individual to the other. And as you can see quite well, the female in the group here often shared with her daughter, but almost never with the male or with the other female. In this group of three females, they also shared selectively. This female shared more with her than with her. And there was food sharing between the two groups, which is really remarkable because harassment avoidance is often suggested as a function of food sharing in chimpanzees. And this is an explanation that can't be applied for these cases. To test whether orangutans share more frequently with close social partners, friends or kin, we conducted a model with relationship quality as a test predictor, and we controlled for food type and repetition and random effects and random slopes. And the model found a clear effect of relationship quality on the probability of food sharing in these orangutan groups. The individual probability to share food with another conspecific increase with the strength of their social relationship, like in chimpanzees. As the proportion of active transfers was higher than known from studies with chimpanzees, we compared our results with those of previous experimental food sharing studies on chimpanzees, and we gained data from a meta-analysis by Adrian Yegi and colleagues and supplemented them by two studies from 2013. We pooled the data on group level and entered each study group as a single data point. And this figure shows on the y-axis the proportion of active sharing in all tolerated transfers. And green boxes indicate the chimpanzee studies, while orange boxes indicate orangutans. And separated for sharing with immatures on the left side and among adults on the right. In both conditions, orangutans shared more actively than chimpanzees. However, we are aware that our study had a small sample size and that the food type used across all studies differed and might, at least partly, have been a confounder. To generalize difference, a systematic study applying the same methods with a large sample of chimpanzees, bonobos, and orangutans would be required. And actually, we have already started this endeavor using the approach of this study with chimpanzees, bonobos, and orangutans. To summarize the results of this study, like chimpanzees and bonobos, zoo-housed orangutans shared frequently when provided with monopolized food, with offspring and among adults, selectively in favor of more closely associated individuals. But zoo-housed orangutans shared more often actively than chimpanzees and bonobos in comparative settings. To conclude, food sharing patterns in orangutans do not qualitatively differ from chimpanzees or bonobos. Monopolizable food sources trigger communication and non-aggressive interaction over food, and food sharing among adults probably serves establishing and strengthening social relationships like in chimpanzees. We therefore suggest that monopolizable food sources can serve as behavioral enrichment for zoo-housed orangutans by stimulating communicative behavior and pro-social interactions. With this, we leave the orangutans and take a closer look at chimpanzees. And in the second part of this talk, I'd like to introduce you briefly to an ongoing project, the Evolution of Brain Connectivity Project, short EBC. And the EBC is a big collaboration between the MPI for Evolutionary Anthropology and the MPI for Human Cognitive and Brain Sciences that aims at linking behavioral variation to variation in brain structures in chimpanzees and bonobos. The behavioral part of this project is led by Catherine Crockford, Roman Wittig, and Daniel Haun. Kathy and Roman are primatologists with an expertise in studying chimpanzees and their natural habitat, especially the Thai National Park in Ivory Coast. And Daniel is a psychologist with an expertise in comparative psychology in both a cross-cultural and a cross-species perspective. And my field of research in this big project is, of course, related to zoos. And the overall goal of this part is to link variation in cognition and behavior of chimpanzees living in zoos and sanctuaries to social and ecological factors they have been confronted during their lives. But before variation can be linked to factors, this variation has to be assessed, and this is what we started to do in the summer of 2019 with my postdoc colleague Sonja Eber and two research assistants, Isabella O'Neill and Peter Nissen. And to assess individual variation and factors, a large sample size is necessary, and therefore we cooperate with a great number of zoos and also sanctuaries. And so far we managed to collect data in 6 German zoos and at Takugama Chimpanzee Sanctuary in Sierra Leone, which resulted in a sample size of 81 chimpanzees with an age range of 1 to 53 years. And here I show you the general design of our data collection. We applied the same methods, if possible, to all chimpanzees at the zoos and the sanctuaries. And we collect and analyze data from 3 main sources. First, we conduct natural observations, focal and group observations to assess for each individual the activity, the sociability, social relationships, gestures, vocalizations, and tool use in daily life. Second, we apply a standard set of cognitive and behavioral tests, like alarm call tests, tool use, food sharing, and mirror tests. Here, we faced the challenge that we needed tests that were applicable under various conditions in different zoos and sanctuaries. And we always used a group setting. We never separated individuals. And third, we tried to learn as much as possible about the past of the chimpanzees in our sample in order to finally determine factors and circumstances in the early and later life of the chimpanzee that influence the onset and development of cognitive abilities, motoric skills, and social behaviors. The data from all 3 sources will be combined and analyzed for each individual to finally reach our goal, namely linking the variation in behavior to factors. As the EBC project is an ongoing project and data collection is not finished yet, in the remaining time of my talk, I will therefore show you only 2 tests and some details of 2 tests, namely the mirror test and the tool use test. And although I can present only preliminary data, I think that a few conclusions that are relevant for zoos can already be drawn. Let's start with the mirrors. What is known so far on mirror self-recognition in chimpanzees, or for short MSR? There is convincing evidence that chimpanzees are able to recognize themselves in the mirror, for instance, using the mark test paradigm by Gallup. But there are also considerable individual differences, and several studies came up with similar results. They found an MSR score of about 30%. The onset of MSR in autogeny is between 2 and 6 years of age. And there seems to be a correlation between MSR and sociocognitive abilities, and therefore we are interested in MSR in our study. Several factors have been suggested that might influence MSR, for instance, age, sex, or rearing history. So in our study, we wanted to determine factors for inter-individual variation, as well as assessing ontogenetic trajectories for MSR in young chimpanzees. And our methods were very simple. We didn't use a mark test. We used spontaneous interactions with a mirror. And as an indicator, we considered mirror-guided self-exploration to be an indicator for MSR. In addition to the classic setup using a large stationary mirror, in our study fixed to a window pane of the enclosure, we introduced a new setup. Several shatterproof mirrors, portable hand mirrors, are distributed throughout the enclosure. In fact, as many mirrors as apes were in the group. And the rationale behind this new method, or the introduction test of this new method, was that we assumed that a lack of motivation to engage with the big mirror or shyness might produce false negatives for MSR, and we expect that the motivation to interact with portable hand mirrors would be higher. Furthermore, small mirrors are easily to use in all zoos and also sanctuaries. At the zoos, we applied both conditions with a sample size of 47 chimpanzees, and at the sanctuary, only the small mirror condition with 33 individuals. We conducted 2 sessions, 1 hour each per condition, and video recorded all interactions with the mirror. And here I'd like to show you some examples for mirror-guided self-exploration, which is considered evidence for MSR. The chimpanzees explore body parts like the face, mouth, or genital region, which they cannot see otherwise. Here with a small mirror. And this is also interesting. And here are the results of our comparison of how the zoo-housed chimpanzees respond to each condition. The y-axis shows the proportion of time spent on mirror-related behaviors of any type, including exploring, manipulating, and social response. The left box plot refers to large mirror and the right to small mirror conditions, and it is easily to be seen that our expectations are met. The chimpanzees interacted significantly more frequent and longer with the small portable mirror than with the large stationary mirror. And this was also confirmed by the zero inflated beta GLMM we calculated. What this figure also nicely shows is the variation across individuals, especially for the small mirrors. Some individuals were less and others much more interested in interacting with the mirror. And here are the same individuals, the absolute data shown with respect to the age of the individuals of the x-axis. And this graph demonstrates that there is no drop in interest with age. Also, the older chimpanzees show a nice variation in their interest to interact with the mirror. Also, our second expectation was met. The chimpanzees showed more and longer instances of self-exploration with small than with large mirrors, which can be seen in the figure. And also more individuals showed MSR evidence with the small mirrors. We therefore conclude that handheld portable mirrors provide a more sensitive measure for MSR than the traditional large mirror setup. So let's have a quick introduction to our two-use test. it is widely known that chimpanzees use a variety of tools, mainly to extract food from, for example, termite mounds or nuts. But the occurrence of particular types of tool use varies across wild populations, and tool use skills vary across individuals. We hypothesize that tool use and tool manufacture skills are positively correlated with opportunities for social learning in early life and opportunities for practicing these skills. To test this, we conducted a tool use task that required stick tool use comparable to termite fishing or honey dipping in the wild. And with this, we assess variation in motor skills, efficiency, functional knowledge, as well as hand preference. And the setup is, as I said, very simple. For each chimpanzee, we attached a plexiglass tube filled with juice to a grid or other structures and provided underneath a standard set of different tools that vary in their efficiency or usefulness. Long, thick twigs with or without brush, two thin twigs with or without leaves, and short, thick twigs with or without brush. Obviously, the short ones are not appropriate at all, and the long, thick sticks with a brush at the end are the most effective. Here are some examples for structures we attach the tubes to. And now I want to show you how chimpanzees of one group differ in their tool use behavior. In the very beginning, you can see on the right a female who is advanced in tool using and modifies the tip of its stick with her teeth to make it more efficient. Others are less skillful. Some are quick, some are slow in inserting the stick. The female on the right is an example for a very slow tool user. But she managed it. So we are still coding the data, applying a very detailed coding scheme, but to give you an idea of the nature of the data, these are the results for the analysis of hand preference in our tool use task for a subset of 42 individuals. And in this graph, we use the handedness index, which ranges between plus and minus 1. Plus 1 would indicate an exclusive preference for the right hand, minus 1 for the left hand. And the nearer the value is to 0, the more ambivalent is the hand use of the individual. And the plot shows HI across the so far coded subsample. And as you can see, with the median is the line of the median near 0 and the given data distribution, our results replicate previous studies. There is no evidence for species-wide handedness as it is in humans. And these are our first conclusions from the two studies that can be relevant for zoos. Small mirrors stimulate explorative behavior, object manipulation, and self-exploration in chimpanzees. There is a strong inter-individual variation with regard to interest in the mirrors across the age classes, and there is no drop of interest in older chimpanzees. Therefore, small mirrors are also suitable as enrichment option for elderly chimpanzees. And third, presenting as many dipping tubes as the number of group members avoids monopolization by dominant individuals and allows lower-ranking chimpanzees practicing tool use. So, and now I very much hope that I convinced you of the following 3 points. Zoos and sanctuaries provide unique conditions for studying behaviors, cognitive abilities, and influencing factors as a complement to field research. Cognitive and behavioral research can provide useful information for zoo management, husbandry, and enrichment programs. And cognitive and behavioral research can be enrichment itself. And with this, I want to thank all the colleagues at the zoos in Berlin, Bremerhaven, Dortmund, Halle, Heidelberg, Leipzig, Magdeburg, Wuppertal, and at Takugama Chimpanzee Sanctuary for their extraordinary support. Furthermore, I want to thank all former and current supervisors, colleagues and students at Freie Universität Berlin and the MPI for Evolutionary Anthropology in Leipzig and all collaborators within the EBC project. And of course, the Max Planck Society for funding the EBC project. And I thank the Serengeti Park for organizing this conference and you all for your attention. And now I'm happy to answer some questions.
So, Katrin, thank you for your presentation. It was really interesting. We collect a lot of questions. Maybe you can choose one of them to answer now, and the other open questions we will discuss later in the breakout session.
Yeah, okay, thank you. I will do as you wish. So I take the first question. Yes, we expected mothers sharing with their infants most, but there was obviously a sharing between adults as well. And I couldn't say that either males and females shared more with each other or sharing among females. It depended on the special relationship and on a special diet. So in one group is also the proportion of sharing, and not only the amount of interactions. So yeah.
Okay.
I hope this answered your question.
Thank you. So then let's go on with the next speaker. I want to introduce Dr. Julia Fischer from the German Primate Center, and she will talk about baboon diversity, causes and consequences. Julia, if you are ready, please share your screen.
Yep. Okay, does it work?
Yeah.
Okay, so first of all, thank you very much for the invitation. I had been so much looking forward to meeting you all in person and see the hamadryas baboons at the Serengeti Park. Now it's all, we're just, going to keep on pushing. So I'm very happy to talk about baboons, my pet genus, if you will, and the diversity, the causes and the consequences. Yeah, so these are the funders, but more important are, of course, the people who do the research. And I'm here, first of all, naming the PhD students who have been involved in this. I'm going to talk about their research as we go on in more detail. And then also Dietmar Zinner and Kurt Hammerschmidt as senior scientists, very experienced, who also supported much of the research that I'm going to talk about today. And then, of course, this is a German-Senegalese cooperation, and if we didn't have this wonderful partnership with the Senegalese park authorities who support this research, this all would not be possible either. So thanks to all of them. So what is the research interest that drives our activities in the cognitive ethology group? So one of the key questions for me is, what is the link between the sociome, so all facets of a society, of a primate society, and social cognition and communication? How are they interlinked? And this research then hopefully allows us to test two key hypotheses. The one is the social brain hypothesis, according to which social complexity favors social cognition. And the other is the social complexity for communicative complexity, advanced by Todd Freeberg and colleagues, according to which if you live in a more complex society, that also favors more complex communication systems. And the model we're using to test these hypotheses are, not surprisingly, baboons, so a member of the genus Papio. And baboons are really interesting. They live all over sub-Saharan Africa and on the Arabian Peninsula. And there are now 6 different species recognized, although there is some hybridization, so they're not good species, but still it's worthwhile, and there is good foundation to distinguish between these 6 different species here. And there are 4 species, the olive baboon, the kinder baboon, the chacma baboon, and the yellow, also termed coqui baboons by Clifford Jolly, a baboon research pioneer for chacma, olive, kinder, and yellow. So that gives rise to coqui. And these species all live in the classical old world monkey female philopatric type of society, where the females remain in the natal group and the males disperse at different stages of their life. So in chacma baboons, really when they're fully grown and in their prime, whereas in yellow and olive baboons, males disperse when they're adolescent. And so the core of the group are really the female relatives. So the mothers and daughters and sisters and female cousins and so on and so forth. And they often have a very clear female dominance hierarchy, and male also have their separate dominance hierarchy. We also knew a lot about hamadryas baboons, social organization, by the pioneering work of Kummer and Angst, and then later Larissa Swerdell is also still driving a hamadryas project forward. And hamadryas baboons live in a very different type of society, in a so-called multi-level society or a hierarchical nested type of society, where these small blobs in the middle are really units, one male units comprised of a male with a small number of females. And then several of these units team up to form clans, and several of the clans form a band. And then if you go to a sleeping cliff, for instance, you might see several of these bands. So there are over 700 baboons on one sleeping cliff. But if you know everyone, then it's highly predictable who will be sitting next to whom and who will be going out in the morning and foraging with whom. And when I started my professorship, now already a couple of years ago in Göttingen, we looked at the map of baboons and we thought, well, that's actually interesting. There is a white spot still on that map. That's the Guinea baboon. We really knew very little about Guinea baboons. And when you looked at the literature, then some people said, well, they're organized like hamadryas baboons, and others said, no, they're more like the coqui baboons or also savanna baboons. So they're female philopatric. And we thought, well, it's worthwhile to go there and check out and try to find out what is going on so that we really get a full understanding of the genus, of the social organization and characteristics of all the members of the baboon genus. And so we went to Senegal, one of the countries, a key country where within their distribution range. And so here Dakar is a very nice and pleasant place to be, always like 26 degrees centigrade. And then you go inland and then it becomes really hot and dry. And in the Neokolokoba forest, this is where we set up camp. This is our camp now. Here again, members of our team. And so we started this project in 2007. It took us about 3 years to habituate the animals so that we could actually start to identify them and follow them on foot. And now we have what I would call a long-term project, although it doesn't compare to the Amboseli baboon project, which has been going on for more than 40 years now. But we have identified in the meantime over 430 subjects in our database, and so we get nice demographic data. Although COVID, of course, we had to close the camp for a couple of months, but now we are back and running again. And in the first years of our research, what we did find out was that the social organization of the Guinea baboons is very similar to that of the hamadryas baboons. So they also live in a multi-level society. We used a slightly different nomenclature compared to the hamadryas. So we call these units, or reproductive units with the males and females. And then units of several, or aggregations of several such units are parties. And then 2 parties, sometimes 3, can form what we call a gang. And then sometimes there are also hundreds of baboons around you, like a sea of baboons. So they're very gregarious, spatially tolerant of one another. So that is similar to what we see in Hamadryas baboons. They're also not territorial, so we put GPS collars on a couple of animals and we looked at their ranging behaviors, and there are no territories. And also when you follow them on foot, when they see another group of baboons, they typically don't care much. So that's all very similar. And that also makes sense if you look at the baboon phylogeny here. This is now genomic information with more information coming in. This fits very nicely with previous research using mitochondrial information. So there is a big and deep split between the southern clade and the northern clade. And the southern clade comprises the chacma baboons, the yellow baboons, and the kinder baboons. And then the northern clade is the olive baboon, Hamadryas, and Guinea baboon. But there is also ancient and ongoing gene flow between different species, famously also between Hamadryas and olive, and between Hamadryas and yellow also. So it's a mess to really, but what the picture that emerges, that this is sort of the key phylogeny, and so Hamadryas and Papio can be considered sister groups. So perhaps at the base of this branch here, there was this shift between from the female philopatric system to the male philopatric system. And that also now with Guinea baboons allowed us to look at one really dominant hypothesis in the last years. Namely, people had always assumed that the Hamadryas baboons have this social organization because they live under arid conditions. So they live in very poor habitats. There are also water is scarce, and therefore people assumed that their social organization was an adaptation to the habitat. But if we compare the productivity of Cementi, our area, with that of Filoha, where this ongoing old study of the Hamadryas baboons is taking place, we see a much higher productivity in our area. And at least for our population, we can refute this idea that this is the current, that the social organization is an adaptation to the current ecological conditions. So we have to think more about what could have caused this shift, and there are ideas about that it has something to do with the range expansion, but that's of course difficult to test now. But what is also clear, if we take a broader picture, if we look at the habitats where all different species of baboons occur, then we see that the variation of the ecological conditions within species is similar to that of the variation between species. So there's really no good map between ecological conditions and social organization or other social characteristics. So the classical socio-ecological model does not really explain well the differences in social organization between species. Okay, so we've talked a little bit about social organization, but there is more, of course, to a social system than social organization. There is the question of who mates with whom, what are the dispersal patterns, and then importantly, especially for my interests, what about the quality of the social relationships between the animals. And what we see is that females are responsible for the gene flow. Males are more resident, if you will, but sometimes males also leave town. And the females, they associate themselves in the Guinea baboons with a male, but they also leave him at their leisure. It seems to be the case in the Guinea baboons that the males have really very little control over what the females are doing. So what you see here in this map, this is just sort of a snapshot over a little more, about a year, a little more than a year. Here there are the identities of the different males, and the color codes are the different females. And for this time period here, you see that some of these relationships were stable. And so, for instance, Sane, he had these 3 females all throughout the entire time of the study period. But there are also other females, like this one here. She moved from Peter to Bar, and then from Bar to Andy, and then this is the famous Annika. She's still around, so she moved from Peter to Ndur, and back to Peter, and then to William, and then to Usman, and then to Bar. And now she has a new young male called Zigzag. So she's been, quite busy traveling around and shopping around for males. Once a female has settled with a male, normally he is also the father of the children. So we have about 98% of the copulations take place with that male, and paternity certainty is quite high. So out of the first batch of infants that we analyzed, about 90% were fathered by the male the female was affiliated with. Overall, there's really very little coercion compared to the Hamadryas baboons, where the males are highly aggressive, where there's a lot of herding, neck-biting females, making sure the female is following him all the time. We see females, even females that are swollen, that are fertile, highly attractive. The male doesn't care if she walks away for more than 50 meters. So females they can also counter-aggress males, so they have a pretty good life in Guinea baboons compared to Hamadryas. And here's just a little film. So what you see here on the right-hand side is a male, and on the left-hand side is a female here. And here there is a bushbuck they had just killed. So they killed them by basically stumbling over them and then starting to eat them alive. And what you will see here is very typical. So she's grooming him now. And now she's presenting, and he's mounting. And now watch what her hand is doing. So here she goes. She takes the prey, and he doesn't even follow her. He doesn't take it away from her anymore. So that is also food sharing, if you will, of a different type here in this species. Okay, so now this is male-female. What about male-male relationships? When we look at male interactions, we see three types. Affiliation is frequent and much more frequent than aggression. The most frequent social behavior are greetings, so highly ritualized stereotype behaviors these males do. I'll come back to that in a moment. And aggression is really, really rare among males, especially decided aggression, where we could say, okay, here's a winner and here's a loser. And the consequence is that we cannot establish a clear dominance hierarchy among the Guinea baboon males. So especially with the new methods that do not only allow you to determine a rank hierarchy, but also the certainty or uncertainty with which you can determine a rank hierarchy. We see here that sort of the key players in the group that we cannot really say with certainty who is higher ranking or not. So there's a huge overlap due to the scarcity of the aggressive interactions. The only difference are here the young males or very old males. They are clearly lower ranking than the prime or primary males. But overall, if you're walking with them, it's very, very difficult to say, okay, here's the top male, the alpha male, and so on. Whereas in the Chacma baboons that I studied before I studied with the Guinea baboons in Botswana, there it takes you about a day to figure out the rank hierarchy because it's so obvious. And so here's, yes?
Sorry to interrupt you, you still have 5 minutes.
Okay. But on my watch, it's actually another 9. I'm only at 16 minutes on my watch.
Okay.
So we started late. We started 5
okay.
minutes late. Yeah. So sorry for this, because otherwise I would really have to rush. Okay, so greetings here. Greetings occur also in other baboon species, except the Chacma. Chacma males wouldn't touch each other, but they have different functions. And in the Guinea baboons, the function is to reinforce party membership. So you see the greetings mainly of the males within a party, and the intense greetings really only among friends. So you can conceive this as a signal of trust, if you will, when they're touching each other's by the penis. But let me now compare a little bit the Chacma baboons and the Guinea baboons. Chacmas, as I said, unilevel, male dispersal, high aggression, territorial, concert pairs between males and females, and friendships during lactation and coercion of females. And here, Guinea baboons, everything is different. Multi-level, female bias dispersal, male tolerance, and so on and so forth. So how are these differences in the types of societies and type of despotism related to social information processing and signaling? And the material I can compare our studies to is the research done, led by Dorothy Cheney and Robert Seifert. And we know from their work that Chacma baboons are extremely sensitive to playbacks where you simulate changes in rank relations, or you simulate the breakup of a consort pair, or you simulate an intruder. And the Chacma baboons are always super interested in such kind of information. And when we started with the Guinea baboons, we thought, okay, let's look at neighbor-stranger discrimination. It's almost like an intruder experiment. So we played the grunts of a male that was either a stranger or a neighbor. And there are two hypotheses here that we wanted to test, that either they respond more strongly to strangers or neighbors. But the Guinea baboons, they were neither interested in strangers nor in neighbors. They were really only interested in grunts of their own group members. So that's where they spent, a lot of time looking and trying to check out what this other guy was doing there. So completely opposite behavior. And another experiment we did recently, this is where we looked at, so a subject, we have, here we on the right-hand side, you see the units in 3 different colors. They have different varying numbers of females, can be anything between 1 and 6. But let's assume here we have these 3 different units, and we have a male from the green unit, and we play to him either a male-female interaction between a male and a female that are not together, or as a control condition between a male and a female that are together in a unit. And we predicted he would be surprised if he would see this interaction between a female that may have transferred, actually. So that was the prediction, but we found again, they were more interested when a male and a female were talking to each other that were in the same unit. So again, we see here an opposite reaction pattern, opposite value of social information in one species compared to another. And that makes sense if you think about the fact that the Chacma baboon society is so despotic, and that if you detect a violation from the norm, it can also have implications or costs directly for you. So therefore, you really want to track what is going on. Whereas in the Guinea baboons, they appear to ignore social noise, perhaps so that's the consequence of their being so gregarious. But at the same time, they have this familiarity basis. So they're kind of reconfirming, apparently, what is already happening. But that begs the question, of course, how do they keep track of changes in their social environment? So we don't know this yet. But we do find in 2 studies now that they're really more interested in already known information. What is really striking if you look at the baboons is irrespective of the type of social system they are living in, and irrespective of the dispersal patterns and despotism, the structure of the signals they're using, both in the vocal domain and in the non-vocal domain, has hardly changed at all over 2.1 million years of evolution, if you will. So the highly conserved structure of the vocalizations, but also the other expressive patterns in the species. And that does not really fit with the social complexity for vocal complexity hypothesis. You would expect that perhaps if you think the Guinea baboons are living in a more complex society, that there would be variation, but there isn't. Okay, and almost coming to the end, let me just look at the function of a signal once more. So here we know from the species, from studies in Chacma and in olive baboons, that females and males grunt when they approach others. And these analyses have also shown that infant handling and affiliation are more likely after grunting. And therefore, Joan Silk and colleagues have termed this as signals of benign intent. So basically, a high-ranking female, when she approaches a low-ranking female with an infant, she needs to grunt because otherwise the other female would walk away. so rank is a very strong predictor. But if you're friends already, you don't need to grunt really, because the other one knows she's not going to bite you. And in guinea baboons, we find overall similar patterns. So grunts are also, if you will, signals of benign intent. But since we don't really have rank, we couldn't use this as a predictor. But what we looked at was the certainty of the knowledge about the relationship. So here we see bond strength on the x-axis. So here are weak relationships, and here are strong relationships. And if the relationship is weak and therefore more uncertain, then the animals, if they were approaching each other, do grunt more to signal, really, I don't want to do anything, I just want to touch your infant. Whereas if you are friends, you don't need to do that so frequently. So we see here that the general disposition is shared between different species, but exactly how they use the calls, that is flexible. That's where the flexibility is. And here we see that the need to signal the benign intent is somewhat relaxed in the guinea baboons. So we see that both a motivational and a strategic component explains signal occurrence. So to sum up, I think baboons are an interesting model to study social evolution. The socio-ecological model does not explain the species differences well. The structure of communicative signals is highly conserved, and different types of societies put different premiums on social information and the need to signal. And the value of different types of social information varies with social system characteristics. So we are basically extending the social brain hypothesis. It's not necessarily so that you become smarter, but what you value, that can vary greatly between different types of societies. But we're not saying the guinea baboons are smarter than the chacma or vice versa. But we see that there is an additional component we have to look at when we study differences difference between different species. Okay, so that's it from my side, and I look forward to your questions.
Thank you, Julia, for this really, really interesting talk. Let me share the question with you. So, yeah.
Yes, that's a question about the relatedness patterns. Yes, so we looked at this, and the males are indeed slightly more related to one another in the parties. I mean, a unit only comprises one male, yeah. So, but the units within a party, there the males are slightly more related to one another, and they're also slightly more likely to be friends with somebody who's related. But it's not completely predictive. You can also be friends with somebody you're not related to. So yes, there is, and we don't really know if there's selection for interacting more friendly or supporting more your bond, your related animals, or is it a byproduct of the male philopatry, so that ultimately just by this process of the female dispersal and males staying behind, you have more related animals, of course, in your surroundings. Certainly the effect is not very strong.
Okay, thank you for your answer. Now let's come to the last talk in this session. I want to introduce Verena Behringer from the German Primate Center and Max Planck Institute. She will talk about cell-mediated immunity between wild and zoo-housed chimpanzees and bonobos. And she also pre-recorded her talk. So, Friederike, can I ask you again to start the video?
Hello and welcome everybody. My name is Verena Behringer, and today I would like to talk about differences in cell-mediated immunity between wild and zoo-housed bonobos and chimpanzees, and the potential implication of animal welfare and conservation in these two species. Environmental conditions such as energy availability and pathogen exposure are affecting health, like parasite infection and survival of an individual. They are mediated by individuals' immunity. All three of them have implications for animals' welfare. For example, they affect immune ontogeny, and means the development of the immune system, and also species-specific vulnerabilities, and finally, healthy aging. They all three have also implications for conservation. like zoonotic disease transmission, or in times of climate change, animals are confronted with new pathogens and species have different adaptive potential to them. This is all considered in the rising field of eco-immunology. Eco-immunology is a framework that estimates costs, benefits, and fitness consequences of different immune defense strategies. It relates this variation in immune responses to phylogeny, sociality, and ecology. It tries to answer how immunity, the capacity to resist a pathogen, affect life history strategies within and across a species. Within this field, the field of macroimmunology exists. It considers habitat and geographical differences in immune phenotypes across population. That includes also variation in adult immunity, as well as variation across immune ontogeny. And maybe both of them, one of them, or maybe also none of them, has many implications for welfare and conservation in the species. Comparing environments is usually challenging, because there are many factors contributing to the differences. However, zoo and wild populations allow to compare two environments that are totally different in terms of resource availability, pathogen exposure, pathogen diversity, and morbidity and mortality risk. To explain it more in depth. Resource availability is assumed to be predictable and unlimited in zoo house individuals. Pathogen exposure and diversity is assumed to be more novel in zoo house animals because they are confronted with a lot of human pathogens, whereas wild living individuals have a co-evolution with their pathogens and are more familiar with them. Morbidity and mortality is assumed to be lower due to veterinary interventions in zoo house individuals and higher in the wild. So why studying this topic in apes? Because apes are a long-lived species and they favor investment into long-term immune strategies with high initial metabolic costs. Also, they have a long developmental period, and this long period allows them to mediate adaptive immune adjustments. That means that they have a larger variation of functional immune phenotypes. My data set consists of two. One is collected from chimpanzees between 2006 and 2014 and includes more than 600 urine samples collected in zoos worldwide, as well as in two African field sites, one in the west and one in the east. The second data set was collected in bonobos between 2008 and 2019 and consists of over 800 urine samples. They were also collected in zoos worldwide and from one African field site in the DRC Congo. Immune responses are measured in our study with neopterine, and maybe you are not so familiar with neopterine, so let me explain what it does. So neopterine is produced by monocytes and macrophages and dendritic cells. An increase in neopterine level reflects the activation of the cell-mediated immune response. This is one part of our immune system. Increasing neopterine levels happen after intracellular pathogen infections. That means after infection with viruses, for example, HIV, measles, mumps, also COVID and influenza. And also after infection with intracellular bacteria such as tuberculosis or intracellular parasites such as malaria. We have measured urinary neopterine in our lab using a commercial neopterine ELISA kit. This kit was previously validated for the use with bonobo and chimpanzee urine. To analyze our data, we have run a generalized linear mixed model for adult chimpanzees, and we have run a generalized additive mixed model for non-adult bonobos. So let me show our first results. On the y-axis, you see urinary neopterine levels. On the x-axis, we have here in blue the urinary neopterine levels of zoo house chimpanzees, and in yellow the two African field sites. We found significantly higher neopterine levels in wild compared to the zoo house chimpanzees. This difference suggests that wild chimpanzees have permanently challenged immune systems. This is maybe due to a higher diversity in the microorganism or a higher disease burden. That also includes that they have to invest permanently in their immune systems. We thought, oh, this is an interesting pattern, but is this generalizable? Is that where does it come from? So do, for example, bonobos show the same difference in neopterine levels between zoo and the wild? And does the environment affect how their immune system develops? Let me show you the result for the bonobos. Again, on the y-axis, we have the log-transformed urinary neopterine levels. On the left side, we have the wild bonobos, and on the right side, we have the zoo house bonobos, in blue the males and in red the females. On the x-axis, you see the age at sampling. What you see here is also that in both environments, we have higher neopterine level at the beginning of life, and then they decline, and then they stay. So environmental conditions did not affect neopterine levels in non-adult bonobos. Wild and zoo house bonobos did not differ in their neopterine levels, and they also do not differ in their immune ontogeny. However, measuring neopterine levels can be used to monitor immune ontogeny in wild and captive populations. May you wonder why we have used non-adults and why we do not have non-adult chimpanzees, but we do not have measured so far non-adult chimpanzees living in zoos. But a colleague of mine, Theresa Löhrig, has measured non-adult wild chimpanzees, and she found that they have on average more than 700 nanogram per mil. This is in comparison to zoo and wild living bonobos twice as high. So wild living chimpanzees have neopterine levels twice as high as zoo house and wild living bonobos. These species-specific differences in neopterine levels maybe reflect that there are differences in the pathogen prevalence. For example, we know that SIV is common and widespread in chimpanzees, but so far no wild bonobos was tested positive for SIV. It is also possible that differences in immune functioning and disease susceptibility exist. We also know from previous studies that bonobos have a reduced MHC class I diversity. MHC codes for proteins which are essential for the adaptive immune system. So bonobos have selected for malaria protective MHC allotypes, but bonobos are maybe more vulnerable to virus diseases than other apes. And I guess this is an important information for keepers, for people who are habituating bonobos, and especially maybe for ecotourism. We already know from a study published in 2012 that some epidemic diseases affect bonobo distribution already. For example, bonobos are absent in areas where sleeping sickness is endemic. And also we know that at the moment, large rivers prevent Ebola from spreading in bonobo distribution areas. In this figure, you see here the bonobo area, and the three other areas represent outbreak areas of Ebola. So with climate change and globalization, it is really likely that bonobos will be exposed to new pathogens. We concluded that species-specific differences in immunity may have causes or consequences for past selection pressures and or the isolation of certain populations. It may limit also species' current spatial distribution and or the colonization of potential novel habitats. Finally, it affects the extinction risk because of shifting disease landscapes with climate change and globalization. Coming to an end, I would like to thank the German Primate Center, the CRC of Antwerp, and the Max Planck for Evolutionary Anthropology. My special thanks goes to all the zoos and the field sites who have contributed urine samples for this project. And finally, I would like to thank you for your attention.
Thank you, Verena, for your interesting talk. Unfortunately, until now, we don't have a question, but if some other questions come up later, we can also discuss it in the breakout session. So completely that brings us to the end of this session. I want to thank the speakers for these really exciting presentations.